morning. This lecture video is going to look at the role, the structures, the function, and the histology of the small intestines, the main digestive organs in our body. Now remember that we have a big lump of food, which we call chyme, that's exiting the stomach through the pyloric sphincter. Chyme is basically all of the food mixed in with the different gastric juices. It's going to leave the stomach and enter the small intestines, where basically almost all digestion is going to occur and where almost all absorption is going to occur. So we're going to break down all of our food into absorbable compounds that our cells can use for energy. All of that absorption is going to take place within the small intestines. The small intestines is about 10 feet long. How crazy is that if you stretch it out or unwound it and stretched it all the way out? It's 5 times longer than the large intestines. It's called the small intestines though because the diameter is significantly smaller than that of the large intestine. The small intestines is divided into 3 sections. The duodenum, this is going to be the section that is connected to the stomach. The jejunum, which is the middle section, and then the ilium, which will be connected to the large intestines. The duodenum is closest to the stomach, like I mentioned. It's a C-shaped curve, and the pancreas is going to sit right inside of here. It receives chyme from the stomach, plus liver and pancreatic secretions. So the liver is going to be able to spit bile into here. The pancreas will be able to spit its pancreatic hormones in there as well. We call the duodenum retroperitoneal because it actually sits behind the peritoneum. We could technically further divide the duodenum into 4 segments: the superior, descending, horizontal, and ascending duodenum, which is pretty amazing because it's a pretty small structure. Looking at the jejunum and the ilium, the jejunum is going to be this middle section. It's about 3 feet, and it runs from the duodenum to the ilium. Basically, jejunum means empty in Latin, and it was supposedly named by the ancient Greeks who noticed it was always empty at death. There's really no clear mark between the jejunum and the ilium, but when we get to the ilium, this is going to be the longest part of the small intestines, which is about 6 feet. It's thicker, it's very vascular, so it has a lot of blood supply to it, and it has a lot more mucosal folds in it than the jejunum. So that's kind of how you can tell whether or not you're in the ilium or the jejunum, is looking to see how many folds there are present. The ilium is going to join the cecum, which is the first part of the large intestines, at the ileocecal sphincter or valve. The jejunum and the ilium are actually like hooked to the abdominal wall by a layer of tissue called mesentery. And it's too bad we don't have the cadavers anymore because you could actually see or have to cut through the mesentery in order to get to the small intestines. Looking at some histology, again, same 4 layers: the serosa, the muscularis, the submucosa, the mucosa layer. But just like the stomach had some parts of these layers, or some of these layers were modified, such is the case with the small intestines. The mucosa and the submucosa layer have specialized structures called circular folds, and also villi and microvilli, and then there are intestinal glands, or glands specific to the intestines, just like there were in the stomach, present as well. The circular folds, you can see these right here, how they kind of fold up, and then the villi is going to be attached to them. The circular folds are deep ridges that are going to be found in the mucosa and submucosa. These folds are really important because they are going to facilitate absorption. So all of the nutrients and all of the smallest particles are going to get absorbed into our bloodstream through these circular folds. Their shape causes the chyme to kind of spiral rather than move in a straight line through the small intestines. And this spiraling slows the movement of chyme and allows the small intestines the time it needs to absorb all of the nutrients out of it. On these circular folds are these hair-like projections called villi, and they're going to give the mucosa kind of a furry-like texture. There are about 20 to 40 villi per square millimeter, and they increase the surface area of the epithelial, which will again allow absorption to take place with greater efficiency. The mucosal epithelial is primarily composed of absorptive cells, which are going to cover those villi. In addition to the muscle and the connective tissue to support its structure, each villus contains a capillary bed composed of one arterial and one venule, so it's got a blood supply to it, as well as a lymphatic capillary called a lacteal. And the breakdown products of carbohydrates and proteins, which are going to be sugars and amino acids, they're going to enter the bloodstream directly from these villi. However, lipids are going to be absorbed by the lacteals and then transported to the bloodstream via the lacteal system. So right here. So sugars and proteins will be absorbed directly into the bloodstream. Fat lipids are going to have to be absorbed by these lacteals, which will then shift them or send them to the lymph vessels. As the name suggests, the microvilli are much smaller than the villi. These are cylindrical apical surface extensions of the plasma membrane of the mucosa's epithelial cells, and they're supported by microfilaments. It is very difficult to image or take a picture of in some way each microvilli, but the massive bristles that they formed, we call this the brush border. And these are going to be fixed to the surface of the microvilli membranes, are going to be enzymes that finish digesting carbohydrates and proteins. It is estimated that there are 200 million microvilli per square milliliter of small intestines. And again, it's going to increase the surface area and allow then, you can see these right here, this diagram, allow for greater absorption. In addition to the three structures that help with absorption, so the circular folds, the villi, and the microvilli, there is also a gland called the intestinal gland, which is also called the crypt of Luberkuhn, which is formed by cells that line the crevices. So they would kind of line in here. You can see where those would be. And they produce intestinal juices. Now, unlike gastric juices, intestinal juices are slightly basic, so alkaline, and they're a mixture of water and mucus. And they are going to be secreted whenever the small intestines start to get descended or stretched out. So again, you can see the circular villi, the circular folds, the villi, and then the microvilli would actually be projecting off of each of those villi. Again, another photo here within each villi. You can see it's got the blood vessels, it's got the lacteal, which is going to be used in digesting fats. And again, this picture here, we've looked at the esophagus, we've looked at the stomach. You can definitely tell the small intestines because of the villi projections that are coming out here. So mechanical digestion that takes place in the small intestines, there is peristaltic contractions, but they're pretty weak, and they're going to be initiated locally by a neural network independent of the CNS. And so if you watch the neural stimulation of digestion video, that kind of explains that. There's the gastroenteric reflex, which is going to be caused by the initiation of stretching of the stomach. And this is going to increase contractions to empty the duodenum. The gastroileal effect, which is going to be triggered by gastrin, and that's going to relax the ileocecal valve so that the contents can go from the small intestines to the cecum, to the large intestines. And in general, it takes about 5 hours for food to move through the small intestines. Chemical digestion is going to be run or kind of controlled by the intestinal juice, which we produce about 1.8 liters per day. It's mostly water from the mucosa by osmosis, but the rest is going to be secreted by intestinal glands. It's activated during the cephalic phase of gastric secretion, so that's when intestinal juices will start to be produced. Like I mentioned earlier, it's considered a buffer because it's basic. It's about 7.4 in its pH, so it helps bring the pH up, because remember that chyme is really, really acidic. And it is the activation of enzymes for digestion. So that intestinal juice will activate the rest of the enzymes needed for digestion. Some of the important hormones that are going to be used or released or secreted in the small intestines or play a role in includes gastrin. It's going to be produced in response to high protein chyme, and it's going to stimulate the production of acid and enzyme. Secretin, which is going to be released in response to low pH chyme. It's going to increase buffers and bile secretions. CCK, which in response to high fat chyme. In the pancreas, it's going to increase enzyme production, and in the gallbladder, it will cause the ejection of bile. And then GIP will be released in response to high fat, high glucose chyme. So depending on what is making up our chyme, will determine which one of these will end up getting secreted. If it's a high fat, we're going to be GIP or CCK. If it's a high protein or high sugar, we're going to be looking again at the GIP. If it's got a lot of protein in it, if we just ate a steak, for instance, then we're going to be releasing gastrin. And again, this is a great diagram kind of looking at how these, the food in the stomach is going to cause gastrin to be released. This increases gastric acid production and motility, increasing the activity in the stomach. Then it's going to go the chyme to the duodenum, which is going to stimulate the release of GIP or CCK and secretin. Those will work on the pancreas and stimulate the pancreas to release insulin or release enzymes and other buffers into the duodenum. And then as we move through, enter into the jejunum, we're going to be absorbing all of those different nutrients. And then there are inhibitory factors that are put in here as well. Digestion in the small intestines. Most important digestive processes are complete in the small intestines. Most are done by enzymes and buffers from the pancreas and the liver. And the small intestine enzymes are going to reside on that brush border of the villi, those microvilli.